Good afternoon, everyone. Welcome to the session. We are still 2 minutes away from starting, so we'll be just waiting for more participants to join and we'll get going. Good afternoon again, everyone. Welcome to this session. We have 52 participants that have joined. We're just going to give a minute or so for others to join as well, and then we'll get started.
I think you're on mute.
Thanks. Yes, I was just saying, if everyone would just bear with us, we're just waiting for more participants to join. I see they're coming in quite fast now, and then we'll get started. All right, I see we are 3 minutes into our time and we have a lot to cover. So I think we're going to get started now. We are recording, so we will be sharing this, the recording afterwards for everyone who maybe missed this introduction. So good afternoon to you all, and thank you for joining. I'm Lucia Skumbi. I'm the Elsevier Research Intelligence Representative in Sub-Sahara and North Africa. And I would like to extend a very warm welcome to this kickoff of the Research Toolbox, Scopus Researcher Toolbox series. The aim of this series is to enable researchers, postgraduates, students, and postdocs to be more efficient and more strategic and ultimately successful in their research endeavors. We have planned 5 sessions for this series, and they cover making sense of open access and preprints. We'll also cover getting the most out of your author profile, responsible use of metrics, a Scopus perspective, and then a session especially for journal authors, journal editors, I mean, which will be on the 25th of November. And that will cover then how to get indexed in Scopus and why. And then, of course, today's session, we aim to kick off the series on a high note, which is why we invited a Cell Press editor to share her insights about how to get published. Cell Press then being a flagship imprint of Elsevier, publishing more than 50 prestigious journals in the biomedical sciences. But before we get to this, I would just like to share some housekeeping and then to also introduce the members on our panel today. So in terms of housekeeping, the session is going to last 1 hour. Our speaker will present for 45 minutes, which we will then, and then we'll leave about 10 minutes then for Q&A. So please ask your questions via the Q&A box, then we'll get to them at the end of the session. The session is being recorded and everyone will receive a link to access the recording as well as the slides. So over to the introduction. We have today is Dineshri Moodley. She's the customer consultant for Core Solutions, mainly covering Science Direct, Scopus, and Mendeley. And then also very importantly, our speaker of today, Rebecca Eccles. She has a research background in biochemistry, genetics, and molecular biology. She completed a PhD at the Max Delbruck Center for Molecular Medicine in Berlin. Rebecca says that she misses the microscope, but as an editor, she loves delving into new and evolving fields, talking science and working with authors to help them best communicate their work, as well as highlighting people and stories behind science. Rebecca is based in London, in the London office, and has been a scientific editor with Molecular Cell since January 2020. So with that, I'm going to hand over to you, Rebecca, and we look forward to the insights that you'll share with us.
Yep, thanks.
Sorry, and while I was presenting, I didn't notice, I wasn't looking at the screen. I also see we are joined by other colleagues, John Sturley, the account manager for South Africa, and Sharif Ghazi, the account manager for Sub-Sahara. Welcome to the panel as well. Over to you, Rebecca.
Thank you. Can you see my slides okay?
Yes,
Yep.
It looks perfect.
Brilliant. Thanks for that introduction. Yeah, and I'm delighted to be here today to talk to you and give you some of our essential tips for publishing in high impact journals and some lessons from Cell Press. So yeah, I don't think I need to go in to my background, as Lucia introduced that very nicely, but just to say I have quite a diverse background in molecular biology, which I think places me very well for this position as a scientific editor at Molecular Cell. So what I'm going to talk to you about today, so just a brief overview about Cell Press and the publishing opportunities that we provide. And then we'll talk about the preparation of manuscripts for publication in high impact journals, and then get into the peer review process. So Cell Press, where did it all begin? So Ben Lewin, back in 1974, launched Cell, the single journal. And today John Pham is the editor-in-chief of Cell. But now we expanded, and as Lucia said, we have over 50 titles, and we're a leader. We've been a leader in life science publishing since the launch of Cell in 1974, and Molecular Cell came soon after in 1997. And then with topic-specific journals like Neuron and Immunity. And we then expanded into the physical sciences, then into the environmental sciences, and recently into the medical sciences. We're really an all science publisher. We also have gold open access journals. So we really have the right option for you. We have a vast range of topics and all different types of publishing options there. So the gold open access journals include Cell Reports Physical Science, iScience, and Helion. And we also have lots of different trends reviews journals. So there's also a reviews journal there for everyone. And we also have partner journals with the societies, such as the Biophysical Journal. And yeah, like I said, we have over 50 scientific journals, and we're now an all science publisher across the life, physical, earth, and medical sciences. And our motto is science that inspires. And we also provide a wide array of learning resources on our website, cell.com. And these aim to enable researchers to maximize the publication of their research. So from the journal homepage, cell.com, readers can freely access galleries such as Cell Press Picture Show, Cell Press videos and podcasts, best of collections, special issues, of which Molecular Cell just published a metabolism focus issue, which we're really proud of. And an author's father actually painted this cover, which is pretty cool. Yeah, so there's plenty of exciting things going on the website, also to promote the articles that we publish. And we also provide mentoring and job connections. So we have Cell Mentor, which is a new online resource that empowers early career researchers with career insights and publishing advice, and also input on experimental techniques. And as trained researchers ourselves, we seek to continually support the collaborative connections between researchers. And we do this by organizing Cell Symposia. And I'm also involved in organizing one of those that's going to be starting on November the 1st for 3 days on biological assemblies. It's unfortunately completely virtual, but I hope that means we can reach a wider audience. But aside from the Cell Symposia and longer conferences that we organize, we also have Lamp Links, which are one-day programs, and Cell Press webinars, which are on scientific topics as opposed to this webinar on authoring. So we're really engaged in the communities that we serve. So just a little bit about the new launches that we have. So Chem launched in 2016. This was the first foray into the chemistry and physics side of things. And then Joule came soon afterwards, and Matter in 2019, and One Earth was the first foray into the environmental sciences even. We then launched the reviews journal, Trends in Chemistry, in 2019, and Cell Reports Physical Science joined the Cell Reports family in 2020. Like I said, we're also now bringing medical science to Cell Press and really aiming to bridge the gap from bench to bedside. This started with Med, our flagship medical journal, which is supposed to be a sister journal to Cell, Chem, and Joule. And Cell Reports Medicine joined the Cell Reports family. And in 2021, we launched Chem Catalysis in June, and all of their content is free throughout this year. And Cell Genomics just actually published its first papers, with the first issue coming on October 13th. So exciting times at Cell Press. We also provide a variety of publishing options, and we're committed to meeting the growing demand for open access and new and different publishing models. So Cell Reports belongs to the Cell Reports family, of course, and Cell Reports Methods is the most recent family member to join. And we also have interdisciplinary gold open access journals such as iScience and Helion. And with iScience, the I really stands for interdisciplinary. We also have gold open access partner journals, such as HGG Advances in the Innovation, And hybrid publication option is also available in all of the Cell Press journals. We also have STAR protocols, which was another recent launch in the open access type of journal. And this is aiming to make researchers' lives easier by publishing robust and usable protocols. And there you have the opportunity to publish your protocols alongside research articles in our other journals. And Patterns is all about the science of data, and that was another launch in 2020. So let's get on to the preparation of manuscripts for publication in high impact journals. So a few questions to ask, to ask and answer before you write. So think about why you want to publish your work. Have you done something new? Is there anything challenging in your work? Is the work related directly to a current topic of high interest? And have you provided solutions to some difficult problems? If you can answer yes to some or all of these questions, then start preparations for your manuscript. But first of all, you need to pick the right journal. So identify the sector of readership for which the paper is meant, and identify the interest of your audience, and make sure this matches the choice of journal. Read the aims and scope page, and look, does the journal publish on this topic? Look at the papers that you cite and do a literature search. Are these papers within the journal? The right journal will also have editors who know the field and the important questions being asked, know the right reviewers, and they can also guide the process and resolve disputes. Informal pre-submission inquiries can be really useful to gauge the interest of a journal and the potential fit at an earlier stage. So you can then tailor the paper really for the right journal. So for example, for molecular cell, you would maybe take the paper more towards a mechanistic point, whereas for cancer cell, you would really emphasize the cancer relevance, for example. So at this point, I'm just going to briefly introduce molecular cell because I'll use it as an example going forward in terms of scope. So we want to publish the best molecular biology, and we're looking for advance really from the mechanistic angle. And we publish a variety of different article types. So as well as our main research articles, we also publish resource and technology articles, as well as short, we also have a short article format, which is intended for concise, highly provocative and fully validated findings. And in addition to this, we also publish review and perspective articles. So why publish at Cell Press? Well, we have full-time in-house professional editors, and we act as impartial surrogates for the broad readership. We actively engage the authors and the reviewers. So we always share reviews between the reviewers and encourage a dialogue and a consensus. So we'll ask, do you think this point is fair? Do you think this point really needs to be addressed? And we also encourage authors to discuss revision plans and help guide revisions. We really highlight what is critical for the journal. We're not just sharing reviews and a long list of points. We're really identifying what is needed to support the main conclusions of the paper. We aim to make timely decisions. So at Molecular Cell, we aim to make that initial decision within 3 to 5 days after submission. So whether to review or to send it back to the authors. And then we aim to make a decision after review around 4 weeks after submission. We're responsive and hospitable. We maintain high scientific standards during the review process, and all of our editorial team decision-making is team-based. We really listen to the communities that we serve. So we're always going to conferences, talking to our advisory board members, talking to scientists and authors about where the fields are going, what's happening, and where the journal should be going. We experiment and we innovate. I think this comes through in our, that we launched STAR methods, which is a part of all of our articles. And we recently also launched a limitation section. So we're constantly trying to improve. We also have a reputation for rigor. So I think STAR methods also nicely illustrates that. And we've also implemented recently new data and code policies. So it's all about ensuring reproducibility, that the work can be built upon. And also at Molecular Cell, we ask for all original data to be deposited corresponding to the figures. And we routinely run image analysis. And then also post-publication, we do a lot of promotion. So we commission preview articles to highlight the work. We also do meet the author profiles, which is something I really enjoy doing. We do author audio and video interviews, and we also have a dedicated press team who will coordinate press releases, and we also promote articles a lot on different forms of social media. So, what makes a good manuscript? It should contain a scientific message that is clear, useful, and exciting. It should convey the author's thoughts in a logical manner, such that that the reader arrives at the same conclusions as the author. And it should be constructed in the format that best showcases the author's material and be written in a style that transmits that message clearly. So what type of manuscript? So as I mentioned briefly, at Molecular Cell, we have different types of articles, and it's the same at a lot of different journals. So articles will be a substantial and significant piece of research. Reports are early communication of significant and original advances, and these are usually much shorter than full articles. So a Molecular Cell, this is 4 figures as opposed to 7 figures for the full article. Could it be a review paper or a perspective? And these should summarize recent developments on a specific topic and highlight important points that have previously been reported, but introduce no new data. Often these are submitted on invitation. So self-evaluate your work and be critical. Is it sufficient for a full article? Ask your supervisor and your colleagues for advice on the manuscript type. And sometimes outsiders can see things more clearly than you. And this leads on nicely to what I would say is one of our biggest tips. So get feedback before a submission. Recruit colleagues outside of your area to review the manuscript and ask for an honest appraisal. So is the question clear? Is the relevance of the research clear? Is the flow of logic clear? Is all the jargon defined? And do the experiments support the conclusions? So as the researcher, you're so close to the work, and it may be obvious to you that pKa means protein kinase A, but is it obvious to someone outside the field? Does it make sense, the flow? And I think really the main thing is the question clear and the relevance of the question clear, and are you answering that question? And if English is your second language, we recommend asking a native speaker to check for grammar and clarity. So just briefly a bit on publishing ethics and your responsibilities. So submit to only one journal at a time. Pre-submission inquiry, like I mentioned, to multiple journals is okay because this is an informal inquiry. Co-submission at 2 cell press journals is an exception to this rule. But rebuttals are still considered under consideration at the journal, so don't submit elsewhere if you have a rebuttal ongoing. So give credit where credit is due in terms of authorship and also citations, and cite related, supporting, but also contradictory work in a scholarly manner. Make your data and code available from the beginning, be completely transparent there, and again, be transparent and complete with methods reporting, including the statistical approach. Make sure you adhere to ethical guidelines in terms of animal and human subject use. Make sure that all data is properly collected, organized and stored, and lab books are properly kept and available, and ensure to include a copy of related papers you've submitted to other journals. And just in terms of the corresponding author responsibilities, so you're responsible as a corresponding author for the entire paper, all of the data, figures, and text. You're responsible for ensuring that authorship is appropriate to the contributions and that all of the authors approve the content and the submission. It's important that you adhere to editorial and submission policies, and that you identify and declare any conflicts of interest on behalf of all of the authors. You should also provide work by any of the co-authors under consideration elsewhere that's related. And you are responsible for archiving unprocessed data and ensuring that the figures accurately represent the original data. And I must really stress here that it's important to check all primary data analysis and figures at all stages, even if the experiments are done in a collaborator's lab. That's still the corresponding author's responsibility. You're also responsible for acting as the primary contact for all communication after publication and for providing any reagents and information that people request. And if readers contact us because they're having trouble obtaining a reagent, we will contact the author and try to facilitate this. So writing a good cover letter. So this is one of the first things that editors will see. So it's really your chance to get the editor's attention and to let us know any additional information that's not included in the manuscript. So what to include, why you think the paper is a good fit for the journal, any additional background that doesn't fit in the abstract, and essentially, or like very important, why do you think the question you set out to address is important, or why is it exciting? Let us know if there's any controversy we should know about, or if there's any competition we should know about. And feel free to let us know any reviewer suggestions and also exclusions. So like I said, the chance to really get our attention and let us know anything additional. But what not to include, the abstract, we already have that. So don't waste any space or time there. And a list of past accomplishments from your lab, we're really judging just based on the manuscript in front of us. So we don't need to know what you published last year or your past CV. Or the meetings you presented, the work at, and the nice feedback you got. This is anecdotal. So when it comes to writing the paper, just some general points. So what's the story? Tell it as simply and concisely as possible. Ensure a logical layout of arguments and flow of experiments. And the chronology of the experiments is not important. And a lot of people make this mistake. They're like, oh, I started with X experiment or A, B, C. And sometimes this isn't what makes the most sense in terms of the story. Make use of summary statements and let us know what you can conclude from the results. And follow journal formatting and guidelines where possible. But in general, we're not super strict about this for initial submissions. So there's no need to reformat between journals as long as it loosely fits and you don't have 20 figures as opposed to the usual 7. And I really want to stress the importance of the title and the abstract. So this is the first thing we as editors and prospective reviewers see, and this is what draws them in to review your paper. And it's also later the first thing that potential readers will see. So when it comes to manuscript preparation, the purpose of publishing a manuscript is to communicate your message to the research community. So if your manuscript is poorly written or presented in a way which makes it the message unclear, there's a risk that the reviewers, editors, and readers may not understand it. So to ensure your work is properly comprehensible, it's essential that you use proper English with concise sentences, correct tenses, and correct grammar. And you can consult the journal's guide for authors for any additional specifications. We also recommend that you ask a native English-speaking colleague to help you edit the final version before submission, or take advantage of one of the many language editing services available today. So, if we break any scientific paper into its main components, this is approximately what it would look like. So the title is a tiny bit on the top, followed by a bit larger abstract or summary, and then the main text of an article. And obviously the main text is the largest chunk, and it's also the most important chunk of any paper, without which, without the main text, the introduction, the results, discussion, experimental procedures, references, there would be no paper. And it's understandable that authors spend most of their efforts on structuring the main text. So why is my top tip about crafting the title and the abstract? Well, they matter, and they matter a great deal. So this is the author's perspective, but what do you think is the reader's perspective? As you can see, it's completely flipped. So the title is the most important there. So use an effective title. So it should be succinct, it should describe key content, the main point should be clear, and it should avoid any obscure abbreviations. It's really there to catch the reader's attention, so you should try to do this in the fewest possible words and be specific and directly reflect the context of your manuscript. Be informative but concise, and again, avoid any technical jargon and abbreviations. It should be easy to understand. It's advisable to discuss the title with your co-authors, and remember that editors and reviewers don't like titles that make no sense or fail to represent the subject matter adequately. You're really setting up the reader of what they're going to see, what they're expecting to find in your paper. So if the title isn't accurate, more importantly, you want the appropriate audience to read your paper. So if the title isn't accurate, the right people may not read it and the community may be unaware of your work. So this could lead to lower citations than the paper deserves. So then, if the title catches a reader's attention, the next thing they check is the abstract. And the abstract is freely available in electronic abstracting and indexing services such as PubMed, Medline, Embase, and Scopus. And yeah, so the abstract should be interesting and understandable, accurate and specific, and brief and to the point. It should be a single paragraph summarizing the problem, the method, the results, and the conclusion. It acts as an advertisement for your article. And yeah, you want to make it as catchy and accurate as possible. And an abstract written clearly will strongly encourage the reader to read the rest of the paper. Many authors write the abstract last so that it accurately reflects the content of the paper, but please ensure you take the time to write this carefully, even if you leave it to the last. So you want to tell the reader what the topic is about or what critical information is needed to move the field forward. And this is your hook. You want to get the reader in. Then you want to describe your key data and how you're addressing this point. Don't list all of your data, but pick the strongest experiments with findings that complement and build up to your main message. And then at the end, you want to link back to the outstanding question, how you solved it, and make your central conclusion. So it's really like a condensed little paper. Yeah, and make sure that relevant keywords are in there too, as PubMed casts a wide net and a big sea. Then the introduction. So the introduction is used to provide context for your manuscript and convince readers why your work advances the field of study. So be concise in the introduction, but give the reader enough information to understand why the work is important. Introductions of reports can be shorter and sometimes only one paragraph. But in general, this section shouldn't be a history lesson, but you do need to introduce the main scientific publications on which your work is based. So cite a couple of original and important works, including recent review articles, to give the reader a sense of how your work fits into the literature. Take care to not make citations to irrelevant references, including those from your own work. Ensure that you have a balanced overview and make sure you clearly address the following. What is the problem you are ultimately trying to solve? Are there any solutions already? If there are, what's the best solution and what are its limitations? And what is your work trying to achieve? Provide a perspective that is consistent with the journal where you're submitting. And in general, though it can be tempting to reuse introductions from your previous papers on similar studies, every paper should present a new result and the work should be specifically motivated. So this requires a unique introduction every time. Then the results section. This is where you describe the important results of your research. So again, be clear and easy to understand. And you should present the results that are essential to the discussion, which means that only data of primary importance should be shared. Data of secondary importance can be put in the supplementary materials. But again, do not attempt to hide any data in the hope of saving it for a later paper or split the results of one project into many papers. This can really dilute the effect of the work and can result in lower quality papers, and you may lose evidence that reinforces your conclusions. So use subheadings to keep results of the same type together. This will make it easier to review and to read, and decide on a logical order of the data that tells a clear story. Use similar headings for figure legend titles as for the paragraphs, and these should be informing us of the result. So protein A interacts with protein B instead of IP experiment showing, or yeah. So these should be really telling us the result. Feature any unexpected findings and provide statistical analysis, and also use high quality illustrations and figures. And in terms of the data preparation, so make use of colors and shapes in figures to highlight appropriate data. Graphs should not appear crowded, so try to present at most 3 to 4 data sets and use well-selected scales and label the axis clearly. And in terms of scales, I would try and keep these consistent throughout and use different symbols to discriminate between data sets. Ensure that you have all proper controls, so molecular weight markers on Western blots and scale bars on microscopy, all appropriate controls within one figure. So if you're doing a knockdown experiment, you have your protein of interest that's there that's knocked down, and also the loading control, and that everything's there together. Think of the reader. So it's obvious to you that you've done the control, but the reader and the reviewer needs to see it. So make it simple and don't split it over the main and the supplement. Keep things together that make sense. The figure legends should be brief, but they should contain sufficient explanatory details so that the figure can be explained without the need to refer to the main text. And always guide readers to specific parts of figures in the main text. So if you're talking about an interaction in the main text, you can then point the reader to figure 4b, for example, which shows the IP experiment. Then tie it together in the discussion. So this is where you interpret what your results mean, and it's the most important section. It's really your chance to sell your data. A huge number of manuscripts are rejected because the discussion is weak or only includes more description of the results. So you really want to get into here, what do the results mean? And make sure the discussion corresponds to and complements the results, but do not simply repeat the results here. Compare other published results with your own and do not ignore work in disagreement with yours. Confront it and convince the reader that you are correct or better. We've had really problematic cases here. If you do miss something critical, then it's either going to, if the editor notices, it's going to get sent back to you so that you can discuss it. And if the reviewer notices, sometimes the reviewer will get caught up on this as opposed to actually assessing the quality of your work. So really confront these kind of things head on and try and convince us why your results are better or why they are correct compared to other findings. And compare published results with your own. So be careful not to use the following: statements that go beyond what the results can support, non-specific expressions such as higher temperature or at a lower rate. Make sure to use quantitative descriptions instead. Don't use or don't introduce new terms here that aren't already defined or mentioned in your paper, but speculations on possible interpretations are allowed. However, these should be carefully explained and rooted in fact rather than imagination. And if you need to revise the results or discussion, this may involve a lot of work and require further experiments, derivations, or simulations. So sometimes you may not be able to clarify your idea just in words because some critical items have not been studied substantially. So make sure your message is complete before you start writing or submit. Then the references. This is where you cite the main scientific publications on which your work is based. So typically there are more mistakes here than in any other part of the manuscript, and it really causes headaches. So remember to still pay great attention here when writing the section, and always ensure that you fully absorb material that you're referencing don't just rely on checking excerpts or isolated sentences. And this is your opportunity to let the editors and reviewers know, and readers, that you've done your due diligence and truly have an understanding of all the relevant work, especially any recent work. Avoid excessive self-citations of your own work and avoid excessive citations from just one single region. Check the guide for authors to ensure the proper format. So is it numbered superscripts or author date notation? And presentation in the correct format is a responsibility of the author, not the editor. And just to note that editors may use plagiarism detection software to validate authenticity. Then finally, the acknowledgements. So ensure those who helped in the research are recognized. And these are to mention those who have helped but are not authors. So feel free to call out any advisors or undergraduate support, any financial supporters or funders, proofreaders, or suppliers who may have donated materials. So you've written your manuscript. So now let's talk about the submission and peer review process. So at Cell Press, we're around 150 professional editors. And so when you submit your manuscript in our editorial manager system, it will come to one of us at the specific journals for editorial review. We'll read the paper, discuss with the team, and decide if we're going to send it for review or reject the paper. And at this point, we may offer you a transfer option to one of our sister journals. If we decide to send it for review, we will choose the reviewers, synthesize the reports, and then editorially review it, and then decide if we're going to reject after review. And again, we may offer you a transfer option here, or if we're going to invite a revision. So if we invite a revision, the authors or you go off and do the revision and address the reviewers' comments, and then it comes back to us. We decide if they've been addressed properly, if we're going to send it back out for review, and then decide if hopefully we can accept the paper, or if it should be rather transferred to one of our sister journals. And then after the happy accept, there's the editing to fit the journal guidelines, and then the final publication. So just a little bit behind the scenes at MolCell and how we do things. So we're 7 PhD trained editors. There's 6 of us here, and we just have a new editor, Jay, who joined us 2 weeks ago. And this is Brian, our editor-in-chief. And we have 4 editorial meetings per week. And I should say we're based, so I'm in London, the rest of the team is in the US, in Boston, New York, and Philadelphia, I think. And so we meet on Zoom. Mega is apparently in Hogwarts here. And there we have 4 editorial meetings a week where we discuss papers, commissioning front matter, and any complicated situations. And we also have frequent informal discussions between ourselves. So there's a lot of Zoom calls, pinging on Teams and chats. And we have one dedicated editorial operations associate. This is Cami. And we also have a dedicated production editor and production team. So every day we look through all of the new abstracts that have been submitted, and we vote on purely based on the title and abstract with no author information. And this is before the paper gets assigned to a handling editor. It's the first thing that we do. And this is us acting as a proxy for the broad audience and general excitement, the fit for the journal, and then the handling editor who will then go on and read the full paper and before bringing it back for discussion. They will then use this information when the paper is assigned and identify what we're looking for and any questions for them reading the full paper. And here we're also thinking about consistency. We highlight if there's anything similar that we've handled in order to keep our bar consistent, even though this is also constantly evolving. So this again highlights the importance of the abstract and title. So yeah, we just see the title here, the abstract here, and then this is an interactive Excel sheet where we can change our votes here and say if we're interested. So there's kind of like an across and thumbs up, thumbs down, and then we make comments here and give questions that we have just from reading the abstract. So there's some situations where we're all in agreement here with the kind of across vote. We need more information. Here we're much more positive, thumbs up. We've never heard of an E3 ligase acting like this, and I was very interested to read this paper, and we did end up going on to publish it. And in this case, we're all unfortunately thumbs down. And I think this is a paper describing a knockout phenotype. And while interesting, if there's not that mechanistic advance there or that mechanistic point, it's probably not for molecular cell. And then sometimes where we have differing opinions, and it can be a real rainbow with up, down, middle, and yeah. So here Brian thought it could be a good resource. Karen was concerned that they don't seem to go further than just identifying protein-protein interactions. I wanted to know if this was all overexpression or if they do validate with endogenous. And April, one of our old editors, wasn't convinced there was anything particularly new coming out of this. So this just gives you an idea of the kind of discussions we're having just based on the abstracts and what we're looking for and how we do things on a day-to-day basis. So publishing is a collaborative effort. The authors send us the final product of their hard work, and editors try to select and improve upon the papers that come to them. And the reviewers uphold the standards in the field and make valuable suggestions to improve the paper. And we're really facilitating communication. So we're really that overlap between all parties, the authors, the reviewers, and the readers. And yeah, the editor's role. So once the handling editor is assigned, so after at Molcell we've done this kind of voting exercise, the handling editor is assigned, we read the paper and then discuss with our colleagues and bring it back to the team. We're assessing the importance of the question and the advance over published literature. And this is really the first thing that comes. And so we decide whether or not to review for the journal. And then if we do review, we manage the review process, invite the reviewers, and coordinate with authors on the revision process. And then we also promote the work at publication. So yeah, what are we looking for? So we're looking for appropriate scope of research. So at Molecular Cell, we tend not to get into things looking at the cytoskeleton, because that seems to be more of a cell biological or better for a cell biological journal, such as Developmental Cell. So we, as opposed to our kind of really molecular scope, and we're really looking for mechanistic advance. So not just phenotypes or description of knockout or a phenomenon. We really want to know what's happening and what's the molecular mechanisms behind it. Whereas cancer cell, for example, or cell metabolism, they would really be looking for in vivo relevance in the cancer or in the metabolism fields. And cell are obviously looking for very exciting new advance over all topics. So yeah, conceptual advance over published literature. We're asking the importance of the question, the technical competence, the clarity of presentation, and also the broad interest. So in terms of mechanistic advance, so say you've discovered phosphorylation of a protein, a molecular cell will be asking these kind of questions. So did we already know this protein is regulated by phosphorylation? Did we know the kinase and protein were linked before? Have you identified a new kinase, a new substrate recognition mode? Is the phosphorylation doing something unexpected? Yeah, and in terms of technical competence, so at Molecular Cell, we'd be looking for things that you're doing work with endogenous proteins, you're using numerous siRNAs, if you're doing knockdown experiments or guide RNAs, that kind of thing. And in terms of broad interest, we're trying to think, is this just interesting for the field? So for me, with like a ubiquitin and degradation background, of course, I'm excited about this ubiquitin paper, but what does the rest of the team think? Could this be more broadly interesting and make conclusions that can be extrapolated to other fields? So then the outcome of the initial editorial evaluation. So we either return the manuscript to the authors with an explanation of why the editors feel it's not likely to be a strong candidate for publication. And if you don't understand the decision, please feel free to follow up in a polite manner. Or we send the paper out for review. And we'll identify the appropriate reviewers, and we also take into consideration authors' suggestions and also exclusions. So please feel free to suggest people. We do listen to these suggestions. We don't want people too close to the field, and we want to ensure that all topics of a manuscript are covered. We check for conflicts of interest, and we also ask reviewers to disclose this. So don't suggest any collaborators or previous supervisors. And if we do ask you to act as a reviewer and you don't have time, then feel free to suggest alternates. So what do we ask reviewers to evaluate? We ask them to look at the technical quality of the data, the degree to which data support the conclusions. We also ask for feedback on the level of interest. So again, to those working in the field, but also to those working outside of the field. Each reviewer assesses the paper from a different standpoint, and we always honor reviewer exclusions. So peer review at Molecular Cell. So a key feature for us is that all of the criticisms and suggestions are anchored to specific points of the paper. We want the reviewer to judge what a paper shows and not what the reviewer wants the paper to show. So this is kind of a guideline that we give reviewers. So we ask them to give a summary and let us know what they think about the advance and if it fits for Molecular Cell, and then to critique, but that these critiques are anchored to all of the main points of the paper. So for each point, we ask them to say if the results are supported or not, and then any other comments. So the editorial decision, we're then integrating all aspects of the paper. We're looking at the reviewer comments, our own assessment of the paper, the nature of the anticipated revisions, and if they're possible, the team feedback and the initial excitement, if that was there. And we do share reviews and go back to reviewers, and we can ask, do you think these experiments are feasible, and do you think they're required or necessary, and do you think this review is fair, or that kind of thing, or would you agree with this point? And we can ask additional experts for further feedback. And we're trying to balance the technical issues with the conceptual interest. And in general, if everything hinges on one result or the results may change, we may reject the manuscript with an option to resubmit. Like I said, we consider whether additional experiments are feasible or reasonable. So is the reviewer asking for mouse models or patient samples, and if this thing is necessary or reasonable? And just to say in general, it's not a simple yes or no tally. So it's not just reviewer 1 says yes, reviewer 2 says yes, reviewer 3 says no. It's really a combination of all of these things. And some comments will be much more critical than others. So when rendering a decision, so how close is the present state of the manuscript to the standard of the journal? And these are then the options. We would accept the manuscript or accept pending minor revisions, which is rare, at least at Molecular Cell. Or we would encourage the authors to respond to the concerns and carefully outline what would be needed from a revised version. Or we might not encourage the authors to respond, providing reasons why we think a revision would likely not be productive. And again, with any revision, we're trying to synthesize the reviewer concerns and highlight what would be critical for us. So when you receive an invitation to revise, read the letter carefully and decide whether any suggested experiments can be completed within the time frame indicated. And if not, consider whether there are different experiments or analyses that could be completed and are aimed at the same question. And I would really emphasize discussing with the editor any concerns on the revision prior to resubmission. So don't just go off piste and follow an alternative storyline. Confirm if the editor would be interested in this. It may be beyond the scope, or without other critical points addressed, it may be irrelevant. So really come back and discuss with your editor. And you can also do this at any point. So it may seem straightforward at the time, but further down the line, you can also come back and discuss, okay, what if we were to do this or this? Revisions, resubmissions, and transfers. So like I said, make your revision count and contact editors with any questions. We're always happy to discuss. And again, at a later time point, if things have not gone smoothly. Resubmission, make sure to include a detailed point-by-point letter addressing the reviewer's critiques. And the manuscript may be subject to re-review by all or a subset of reviewers. And I would say at Molecular Cell, it's probably the norm that your revision would be looked at by all original reviewers where possible. Of course, there's some exceptions. Just a comment there, I would really make sure you include the entire reviewer comments. The context is really important and you don't want to give the impression that you're hiding anything. And then in terms of transfers, these can be requested by the authors. We can contact editors of different journals. As authors, you should also feel free to reach out and before making any transfer. And the manuscript, but also the reviews will be transferred, and then the reviewer identities. And then, this transfers is about giving you options and hopefully helping you to publish as quickly and as smoothly as possible. So upon receiving a negative decision, read the letter carefully and assess the basis for the decision. If the decision's unclear, again, feel free to contact the editor for clarifications and guidance. Dialogue is encouraged, but take time to think and take a step back and reread your response and ensure the tone is appropriate and polite, even if you're angry. Consider transferring to another CellPress journal. And if you do appeal the decision, provide a point-by-point response to the reviewer's concerns and stick to the scientific issues. Indicate how issues could be addressed experimentally and be reasonable in assessing the situation. Is it actually possible for you to do this work? And is it in yours or the first author's, last author's best interest? We may return to reviewers for guidance. So again, make sure the tone is appropriate and polite. And we may also enlist new experts for advice. So again, here, really stick to the scientific issues. Don't tell us, reviewer 2's an idiot, you shouldn't listen to anything they say. Just make it clear with the science and with your scientific arguments. And then the transfer process. So like I said, the goal here is to help you find a place to publish your paper within CellPress as quickly and as smoothly as possible. So this is an author-driven process with editorial guidance. It can take place whether or not your paper was sent out for peer review at the initial journal. And if the paper is transferred after peer review at the original journal, the reviewer reviews and the reviewer identities are transferred. And the authors do have the option to revise the paper before transferring. And again, you can contact editors at the next journal with any queries, and the original editors would also usually be happy to advocate for the paper. So after acceptance, authors and editors celebrate, but again, the work isn't quite finished. So you need to organize the final files according to the instructions and the final resubmission checklist. And then there'll be copy editing, proofs will be shared, and page layouts. And then finally, you'll be given an online and issue publication. And we will, like I said, promote articles through commissioning preview articles, meet the authors, author audio or video interviews, press releases, through our websites and through social media. So if you would like more information on our journals, please visit cell.com. And if you would like to recommend your library to subscribe to specific journals, you can do that by visiting cell.com/recommend, and we'll share this information with your library. So yeah, I think that's all for me for now, and I'd be happy to take your questions.
Thank you so much, Rebecca. That was really a wonderful, practical, behind the scenes perspective. I see there are quite a few questions, so I'm going to hand over to Dineshri to share the questions with us.
Thank you, Lucia. I noticed a lot of questions in the chat about detailing the article publishing process. So I'm sharing 2 of the last webinars that we had hosted, where the process is defined in a step-by-step manner so that you can get benefit from that. If anyone is interested in that, you can get it from the chat box. Both the recordings have been shared. And then specifically for our presenter, we have a question. Can you explain further what you mean by pre-submission?
Yep. So this is an early stage inquiry about whether the journal would be interested in the manuscript. So at CellPress, there's 2 ways to do this. So through our journal websites, there's kind of like a submission box where we ask for different information. So the title, abstract, you can share the figure titles, and just a brief description of why you think it would be good for Molecular Cell. Or you can just email the editors or the journal inbox, and yeah, you can share as much or as little as you would like. So you can really do it earlier on in the process if you would just like to know okay, would Molecular Cell be interested in this topic? And it's a chance to get kind of early feedback. So if you are ready to submit, you can do this to multiple journals at the same time and maybe save yourself some time. And we might say, we're not sure, we need to take a look at the whole manuscript, or we might say, no, unfortunately, it's not a fit for Molecular Cell, or we might say, yes, we'd love to review this, please go ahead and submit. So it's kind of a pre-submission inquiry letting you know if it's worthwhile to submit or not. But you might also get some, if you do this at an earlier stage, you might also get some useful feedback. So we might tell you, we're interested in this particular part, but it would need, it's unclear from the information you gave us, but we'd be looking for this or this. So yeah, I hope that answers your question. So it's kind of getting an answer without doing a full submission. Yeah.
Yes, okay. And the next is, should one always write a cover letter to a journal, or does it depend on the journal?
Yes, I would always say yes. So always write a cover letter, always tailor it for that journal. So you're really pulling out the points that are interesting for that journal. So for Molecular Cell, you're highlighting the mechanistic points and really writing it to like Molecular Cell. Yeah, I would always recommend writing a cover letter.
Thank you, Rebecca. And how many issues does Cell Press publish per year? And what is the manuscript rejection rate?
Oof, that's a good question. So it really varies from journal to journal. So at Molecular Cell, we publish roughly every 2 weeks. And Cell publishes every week, I think. And but this varies. So some journals are only online. So Cell Reports and iScience are only online, so they're constantly publishing new material. And some of the journals like Cell Metabolism is just publishing 1 issue a month. And again, the rejection rate will really depend on the journal, I would say. So I don't have any numbers for that, but I think Molecular Cell, we aim to publish around 300 articles a year.
Thank you. When you refer to do not use too many references, could you elaborate a little bit more on that? And what happens if research is on some literature or conceptual research?
So in terms of not too many references, I guess it's every reference should be there for a reason. So it's not just referencing your own work just so that it's in there. It's every reference should be there for a reason, and should be connected to a point within the manuscript. And what was the second part of the question? If the work's more conceptual.
Just trying to bring this question up again. It just asked for what happens if research is on some literature or conceptual research.
I'm not sure I really get that question. Sorry.
I think that person can maybe send us more details on the question. There are some questions in chat about predatory journals. And I think if you check out part 2 of the recording that I shared in chat, you will get information on predatory journals. So the next question is, as editors, do you check the plagiarism, or is it up to the author?
So we do have software that checks for plagiarism as part of our editorial manager system. Yeah. But it is also up to the author. So this is kind of a fail-safe.
And how do you anonymize author before the paper is sent to a reviewer?
So the authors aren't anonymized. So in our system at the moment, it's only the reviewers that are anonymized. So yeah, it's just whenever we vote, that initial reaction is purely based on the science. and we don't know who is submitting. And then, so that's to try and keep us fair, that we're not biased by big names. But yeah, when it goes out for review, the reviewers do know who the authors are. So there's been discussion about double-blind peer review, but I don't think any of the Cell Press journals have tried this so far, but there has been some talk about doing it.
And is peer review restricted to PhD holders?
I guess not strictly, though in general we would reach out to group leaders to review for us. But what I would recommend is really asking your group leaders, asking your bosses, your professors, if you can help out with the peer review, because we do have a box where they can indicate if they've helped, and I think this is a great training experience. So especially if you want to be an editor, I think it's the more you can read and analyze other people's work is a great exercise. And I should say one of the journals that I highlighted, STAR Methods, they're looking for postdoc and PhD student reviewers because they're really looking for people to review methods, and these are the people that are really using them. So if you do want to get some experience in reviewing, I would definitely tell you to check STAR Methods out and register as a reviewer with them.
There's just 2 more questions. I know we a bit over time. It should be quick ones. So the next question is, if a journal specifies the abstract to be 150 words and the abstract is longer and it's not possible to trim it down, is there any way that you can submit that abstract?
So I'm not sure on the logistics with our systems, but I think for the initial submission, it's not such a big deal. So us as editors, we're not taking that kind of thing into context. We're just looking at the quality of the work, and as long as it's well written and it emphasizes the main points, that's fine. Of course, it shouldn't be too long because readers and reviewers are going to get bored if it's too long. But in general, the kind of cutting down the text to the right length in general can come at later stages. But I have to say, I'm not sure if our software would only allow you to even paste in 150 words. I'm not sure about that. But I think that you might be able to get away with a bit more.
Thanks. And the last question is, can you please advise if it is important to address the main comments of the reviewer to the editor, or just focus on responding to the comments one by one?
So it's definitely the most critical to respond to the points one by one. You always should do that. But I think if the editor has given you a list of points that are critical to come back, I think it's always worthwhile to say we've focused on the points you highlighted, and we've done not in depth, but you can say briefly we've addressed all of these points in this manner, and please see the full response. So the editor will always look at the full point by point response.
Well, thank you so much, Rebecca. I think I'll hand back over to Lucia to close. That was the end of the Q&A.
Thank you.
Thanks a lot for that. And then with this, I'd like to thank you so much, Rebecca, for sharing your experience in a very informative, open and eloquent way. I'm sure the audience learned a lot. I learned a lot today. I thought it was very interesting. So we really appreciate your effort in doing this wonderful webinar for us today. And then I'd just like to remind the audience that we have our next webinar will be on the 14th of October. And then we'll be talking about open access and preprint. So please join us for that webinar as well. So with that, we'll close the session. Thank you very much again for joining, and see you next time.
Thank you. Bye.
Bye-bye.